Welcome to today's Explainer. We're diving straight into one of the most fascinating and, honestly, kind of devious processes in all of biology, viral replication. But I want you to think about this not just as a standard biological function. Think of it as a microscopic heist. It's this highly coordinated operation where viruses, because they literally can't reproduce on their own, break into our cells and totally hijack the machinery inside to print copies of themselves. Understanding this is absolutely crucial for figuring out how diseases spread, how they evolve, and ultimately, how we stop them.
Okay, let's take a quick look at our roadmap. We'll start with the ultimate cellular intruders, map out the 6 stages of replication, profile their different strategies, ask whether they destroy or go dormant, and finally, look at how we stop the viral takeover.
Section 1. The ultimate cellular intruders. Blueprints without a factory.
So you might have heard viruses called obligate intracellular parasites. That's really just a fancy scientific way of saying they are blueprints without a factory. Unlike bacteria or other organisms, a virus literally cannot metabolize or reproduce on its own. It is completely 100% dependent on finding a living host cell to do all the heavy lifting. But, even though they lack a factory, they definitely don't show up empty-handed. They bring a highly specialized, incredibly compact toolkit to the heist. At their core, they carry genetic material, either DNA or RNA, which is the actual blueprint. This gets protected by a tough protein coat called a capsid. Some viruses even wrap themselves up in a lipid envelope, almost like a disguise stolen from a previous victim. Add in a few specific viral enzymes to help pick the cellular locks, and boom, it's exactly what they need to initiate the takeover, and nothing more.
Moving to Section 2, the 6 stages of replication, executing the cellular takeover.
All right, here is the master plan. Every single virus has to navigate this 6-stage cycle to successfully hijack a cell. It always starts with attachment, followed by penetration, where they actually enter the cell through mechanisms like membrane fusion or endocytosis. Next is uncoding to release the payload. Step 4 is biosynthesis, the actual manufacturing phase. Then they assemble all the new parts, and finally, release, where the new variants burst out to infect other cells. Let's zoom in on step 1 for a second, because attachment isn't just randomly bumping into a cell, it's about extreme precision. The virus has to find the exact right receptor molecule on the surface. It's literally like finding the exact right key for a lock. This completely explains host specificity and tissue specificity, why certain viruses only infect humans, and why, for example, SARS-CoV-2 specifically targets the ACE2 receptors on our respiratory cells. If the lock doesn't match the key, well, the heist is over before it even begins.
Skipping ahead to the real climax of the heist, Step 4, biosynthesis. Once the virus unpacks its genetic material, it completely takes over the cell's control center. It forcefully tells the host to stop its normal jobs and instead start replicating the viral genome and synthesizing viral proteins. The virus hijacks the host's 3D printers, the ribosomes, to manufacture thousands of copies of its own parts. The cell basically becomes a reluctant virus factory. Wild, right?
Section 3. Different viruses, different strategies, profiling the microscopic intruders.
Now, not all viruses pull off this heist the exact same way. There's actually a stark contrast in their target vaults. DNA viruses, like herpes viruses or adenoviruses, usually strike the deepest, most secure vault in the cell, the nucleus. They transport their blueprints all the way in to use our own DNA replication machinery. RNA viruses, on the other hand, like SARS-CoV-2, poliovirus, or influenza, they prefer a quicker approach. They generally stick to the outer workspace, the cytoplasm, where some of them can use their RNA right there as instant instructions for the ribosomes.
But wait, there is a third, incredibly sneaky category of virus that flips the biological script entirely. We usually think of instructions flowing strictly from DNA to RNA to protein. So what if an intruder reads the instructions backward?
Enter the retroviruses. HIV is the most infamous example here. They bring this remarkable molecular tool with them called reverse transcriptase. It literally allows them to take their RNA genome and translate it backwards into a DNA intermediate. Once they pull that off, they actually integrate their newly minted viral DNA permanently into the host cell's own genome. The host cell unknowingly reads the viral DNA just like it's its own.
Depending on the intruder's profile, they might pack a whole set of highly specialized lockpicks. You've got DNA polymerases to copy DNA, RNA-dependent RNA polymerases to copy RNA, integrase, which is basically a biological stapler used to insert viral DNA into the host genome, and proteases acting like molecular scissors to assemble everything perfectly during maturation. These enzymes are absolutely critical for bypassing the cell's natural defenses.
Section 4. Destroy or go dormant. The getaway plan.
So the getaway generally takes one of two forms. First is the lytic cycle, a violent smash and grab. The virus rapidly replicates, fills the cell with new virions, and then totally ruptures or lyses the cell, destroying it immediately so the new viruses can spread. But the alternative is the lysogenic cycle. Here, the virus acts as a sleeper agent. The viral genome integrates into the host and just goes dormant. Every time the host cell divides, it unknowingly copies the viral DNA too. It allows the virus to hide quietly for years before eventually switching back to attack mode.
Section 5. Stopping the viral takeover. How biotechnology fights back.
Fortunately, we've mapped out a pretty amazing counterattack. Because we know exactly how this timeline works, scientists can design specific antiviral drugs to block the virus at every single vulnerable stage. If we block the receptors, we stop attachment. If we inhibit fusion proteins, we stop entry. By targeting viral polymerases or reverse transcriptase, we grind replication to a dead halt. We even use protease inhibitors to mess up the assembly stage and specific drugs to block the viral enzymes needed for release. We literally use their own specialized toolkit against them.
But we are building towards something even more incredible. Because beyond just stopping the virus, we're now actually reprogramming these natural hijackers. In biotech, we use modified viruses as vectors for gene therapy, safely delivering therapeutic disease-curing genes right into patients' cells. We're using their replication codes to design revolutionary RNA and viral vector vaccines. We've taken the ultimate cellular intruders and turned them into our most precise cellular delivery vehicles. I'll leave you with this final thought for today's explainer. It's such a profound shift in perspective. As we continue to master the exact mechanics of viral replication, every enzyme, every receptor, every cycle, the very invaders that have caused some of humanity's most severe diseases are rapidly becoming our most powerful tools for healing. If viruses are nature's ultimate hijackers, how will we engineer their perfect machinery to deliver tomorrow's ultimate cures? Thanks for joining me, and keep questioning how our microscopic world works.